So Amino Acids, Proteins and DNA 2018. So what have we got here? You're asked to draw the hydrogen bonds in. So obviously a hydrogen bond bonds between the delta plus on an NH and the lone pairs of electrons on an O. So you just join those up. It would be nice to put delta plus on the H of the NH and lone pairs of electrons on the O, but you don't actually need to for the mark. Okay, explain how these hydrogen bonds form. So the delta plus on the H attracts or bonds to the lone pair of electrons on the O, forms a hydrogen bond. And if you're asked why you get a delta plus on the H, it's because N is very much more electronegative than an H, therefore you get polarity, a delta plus and a delta minus, delta plus on the H and a delta minus on the N. Sulfur to sulfur bond is a bit of a strange one. It happens between two amino acids, two cysteine amino acids. If you look at cysteine, it's got an SH, and so the SH of one molecule bonds to the SH of another molecule. The H is released and it forms an S to S bridge called a sulfur bridge between two amino acids on separate nucleotide chains or within the same nucleotide chain to make a secondary structure. What have we got? We've got a polypeptide chain influences a three-dimensional structure. A three-dimensional structure is a tertiary protein. Draw the structure of the zwitterine of a dipeptide formed between alanine and serine. So what you've got to do is you're going to take the alanine and the serine, and first of all you've got to join those two together with a peptide link. That's a dipeptide. Dipeptide, two amino acids coming together. Tripeptide, three amino acids coming together. So you join them together. The COOH and the NH2 make water to leave a CONH peptide link. Condensation reaction releasing small molecules such as water. You can break that peptide link by a process called hydrolysis, by the way. To add the water back in, you will need concentrated hydrochloric acid to help break that peptide link. But we've formed it now, and therefore we need a zwitterine. A zwitterine is where you take one of the COOHs and you turn it into a COO minus, and you take one of the NH2s and you turn it into an NH3 plus. That happens at a certain pH called the isoelectric point, where the COH releases an H plus around the NH2 on another part of the molecule to form NH3 plus. Straightforward one this. We've got a nucleotide with bases A, C, G, T, C, which stand for adenine, cytosine, guanine, thymine, and another cytosine, and you've just got to match them up. So you've got to know that adenine joins the thymine and guanine joins the cytosine. So you've got to match up what 1, 2, 3, 4 and 5 are. Adenine would join to a thymine, cytosine would join to a guanine, guanine to a cytosine, thymine to an adenine, and cytosine to a guanine. So they are TGCAG are the identification of the bases 1, 2, 3, 4 and 5. You add them up. So we've got an adenine to a thymine is 3 hydrogen bonds, so we're adding up the number of hydrogen bonds. A cytosine to a guanine is 2 hydrogen bonds. A guanine to a cytosine is another 2 hydrogen bonds. A thymine to an adenine is 3 hydrogen bonds, and a cytosine to a guanine is 2 hydrogen bonds. You add them all up and you end up with 13 hydrogen bonds. Then you've got to show a nucleotide. A nucleotide's got 3 parts. It's got a phosphate group, it's got 2 deoxyribose, and it's got a base. Now it's already drawn the base on by the letter, represented by the letter A. It's not a real base, we don't know what it is, but it's already drawn on. You've got to draw the phosphate on, so you've got to realise that the bottom part, the phosphate, the OH, bonds to the CH2OH on the top left hand of the 2 deoxyribose and releases water. And you can either draw another phosphate on at the bottom, or you don't need to, because a nucleotide is a phosphate, 2 deoxyribose and a base, but you do need to show that that bottom OH will have a phosphate group joined to it, and it will lose its OH, so it will lose its H as it bonds to the OH of another phosphate that you don't need to show. You can do if you want to, but you don't need to show it, but you do need to show that where the other phosphate at the bottom will join on, and you represent that by losing an H off the OH as it bonds to the OH of the phosphate to release water.